Hi everyone, my name is Angel Jose, and I'm an undergraduate student at the University of Texas, Rio Grande Valley. And this past summer, I had the opportunity to participate in the USDA REEU Summer Research Internship at the University of Arkansas in the Department of Entomology and Plant Pathology. So I worked under Dr. Rupesh Karyat's lab, and I worked under the mentorship of Alejandro Vasquez Macano, and I also collaborated with Dr. Lewis's lab in University of Nebraska. So my internship was focused on insect and plant interactions, and we specifically looked at crop protection and sustainable pest management using sorghum. So the research all took place in Fayetteville, Arkansas, where we worked in the greenhouse, the lab, and the growth chamber settings. So the goal of the internship that I was in is to essentially just provide hands-on research experience in agriculture to students who don't normally have the opportunity to do so. So I feel like this internship really improved my scientific and technical skills, and I definitely developed like experimental design skills, experience with insect rearing and plant maintenance, data collection and analysis, and also experience in using different types of machinery like the volatile systems and the Leica machine, and also like root imaging software. Throughout this experience, I also gained scientific communication skills, like creating a poster and presentations, and more importantly, how to troubleshoot experiments, maintaining consistency, and working with a large team. On a personal level, I definitely think I became more confident working by myself and better in problem solving and adaptability, and more comfortable in asking questions and collaborating with a larger team. So additionally, the research also reinforced my passion in tackling public health issues. So starting off with my poster mainly focuses on exploring the potential of brown midrib in sorghum in managing sorghum aphid. So sorghum is a really important crop used worldwide. Here in the US, we're actually the largest producer of sorghum itself. However, a lot of its yield is often affected by pests. So we look specifically at sorghum aphid. And so the plant itself uses different defense mechanisms, both direct and indirect, like wax and volatile organic compound emissions to defend itself against these pests. So we looked at BMR, which is a specific gene inside sorghum, which affects the amount of lignin found in the plant. And through this project, we looked at how this gene in a plant can affect how much sorghum reproduces on the plant. So we used three different genotypes throughout this entire experiment. We use RTX 430, BMR 12, and 11A, which is the control genotype, our mutant one, which is the reduced lignin one, and then 11A, which is overexpressed. So all of these experiments are used using these three genotypes. We used different methodologies throughout the entire experiment, whether it came to counting physiology, the organic compound, wax content, finding the root comparison and everything. But here I have two pictured. This is wax extraction and volatile collection. So wax extraction was done by collecting all these three plants, cutting out 8 small circles from each plant, and then placing it in a small Eppendorf tube. We then used 2 milliliters of chloroform and then vortexed the tube, and then we left it out in a fume hood for over the course of 24 hours to allow all the chloroform to evaporate. And what this does is this essentially just leaves the wax from the top of the plant in the sides of the tube. We then waited to find the total amount of wax on the plant. Another methodology I have pictured here is volatile collection. So I thought this was really cool. It's a glass dome placed on top of the plant, which we then push air through, and then we pull air through the top. So that collects all these volatiles, and volatiles essentially just mean smells that the plant gives off. So when you mow the grass and you smell that weird smell, that's a specific smell that the plant gives off, saying like, help, I'm being attacked. So all of this is volatiles that the plant sends off. We collected all these smells in a small little tube, which we then send for further analysis. So the first thing that we looked at was germination rate. So we planted all these 3 genotypes and we let them grow, and we measured this and we counted the amount of days it would grow. And as you can see here, BMR 12 actually has a lower germination rate compared to the other 2 genotypes. This can be a bad thing as it does have less access to resources, minerals, and water efficiency, but it can be looked at a good thing if you look at when predators come, it hasn't germinated as fast as the other ones. The next thing that we looked at was physiology, and this was done using the Leica machine. We looked at different things like stomatical conductance, transpiration rate, CO2, and here It's a little small, but the first color you see here in the darker pink is without aphids, and the second color is with aphids. So if you can see here, aphids actually significantly lower the amount of physiological traits that a plant has. So specifically somatical conductance and CO2 transpiration rates. The next thing that we looked at was aphid count. So this was done by placing 10 aphids on a plant and then allowing it to reproduce over the course of 10 days, which we then went back and counted. And this is honestly the most significant thing in my entire project. BMR 12, which is the middle one here, reproduced the least amount, or in fact, less than half of the other two, the amount of aphids after the course of the 10 days. So this is extremely significant, as this means that there's something in this plant that is fighting off the reproduction, or it's not allowing the aphids to reproduce as fast. We looked at wax content, as I explained before. This was not significant, but we felt the need to include it as it is a direct defense from the plant. The next thing that we looked at was volatile organic emission, compound emission. And these top two graphs here have the mean and the total number of VOC emissions that the plant emits. As you can see here, BMR 12 actually emits a larger amount of compounds compared to the other two. This can be what deters the amount of aphids from reproducing. It could be different reasons. Down here, you can see the amount of aphids, how aphids affect the number of compounds that is reproduced. So green is herbivory, which means after aphids have reproduced on a plant, and then undamaged, which is without aphids. So aphids actually have a significant amount of effect on the number of compounds that's emitted. Here it's pictured the push and pull system with the glass dome on top of the leaf. One of the last things, or one of the second last things that we looked at is the root comparison. So we took the root, we cut it off, we washed it, and we put it into this machine. It's essentially like a scanner for a printer. You put the root in water and it scans the top of what the leaf is, and then it kind of calculates the amount of roots, the tips, the fork tips. And here, this pictured is BMR 12, and this is the regular genotype. So this is something you can see visually. The BMR 12 has a lot more roots and a lot more root tips, which means it has a greater water efficiency. And then here, pictured in this graph, is the number of root forks, which BMR 12 has a greater significant amount compared to the other two genotypes. This can also be a little bit off compared to with the amount of sample size that we used. And also, when you're taking the root out of the soil once you grow it, it is very obvious that we lose a lot of these roots into the ground. So I think for a better analysis, a larger amount of sample sizes could be used. That's everything from my project. If anyone has any questions.